Hi, I am Shalini from Mahek Sharma Group at Isar Mohali, India, and today I'll be presenting our recent JCB work, Rufi1 binds RLHB and mediates endosome to TGN CM6PR retrieval for cargo sorting to lysosomes. So lysosome receive cargo via endocytic pathway, autophagic pathway, and another pathway toward lysosome involves the biosynthetic transport of lysosomal hydrolases. These trafficking pathways are regulated by small GTP binding proteins of RAPs, ARF, and ARF-like GTPases that localizes to different endocytic compartments, and these small GTPases they act as molecular switches by interconverting between GTP bound inactive form and GTP bound active form. So when they are active, they recruit effector proteins to regulate cargo sorting, cargo packaging, vesicle motility, vesicle tethering, and fusion with the acceptor compartment. So our lab studies RL8B, which is a lysosomal GTPase. RL8B binds to its effector protein skip, which binds to kinesin to mediate the peripheral motility of lysosome. RL8B also regulates the fusion of lysosome with other compartments. So RL8B regulates the recruitment of tethering protein complex hops to mediate fusion with the late endosomes. RL8B also binds to plechem1, which binds to LC3 on autophagosome to mediate tethering of lysosome with autophagosome for cargo degradation. So in our work, we have identified a new interaction partner of RL8B, Rufi1. So Rufi1, like skip and plechem1, contains a run domain, and Rufi1 has two isoforms in the cell. Rabe4' is the longer isoform, which has 108 amino acid extra as compared to the shorter isoform, and both interacts with RL8B via its run domain. Rufi1 and RL8B localizes on early endocytic compartments, positive for RAF14 and EA1, and they are negative for late endocytic markers like LAB7 and lysotrachodine. Interestingly, these compartments also contain cation-independent mannose phosphate receptor. To understand the relevance of interaction of Rufi1 and RL8B, we did RL8 knockdown and stain for Rufi1. Rufi1 appears less punctate and cytosolic in RL8 knockdown, which suggests RL8B regulates membrane localization of Rufi1. To find how it is regulating the membrane localization, we did a purified protein interaction assay. And in this assay, we found that the binding of Rufi1 with MVP RAF14 increases in the increasing concentration of RL8B, which suggests that RL8 binding promotes its interaction with RAF14. So this is our working model that Rufi1 initially is in auto-inhibited state. Binding to RL8B relieves this auto-inhibition and strengthens the interaction with RAF14 and its membrane localization. Rufi1 in the previous literature has been shown as as a sorting platform for multiple cargoes. So from this endosome, receptors are either recycled to the plasma membrane. Also, this protein Rufi1 regulates the sorting of cargo towards specialized compartment like melanosomes. Rufi1 interacts with RAF4 and AP3 to regulate the segregation of melanosomal cargo from lysosomal cargo towards melanosome. Interaction with RL8B now poses a new question: how Rufi1 is regulating the late endocytic compartments. For that, we did Rufi1 knockdown and then assessed late endocytic compartment, and we found that late endosome and lysosomes are highly enlarged and vacuolated upon Rufi1 depletion. Our experiments also shows that there is a delay in the trafficking of lysosomal hydrolases in Rufi1 knockdown. So we looked at the receptor of lysosomal hydrolases via CD8M6PR trafficking assay, where we express the CD8M6PR chimera in HeLa cells and then surface labeled the receptors by anti-CD8 antibody and chased it till 60 minutes where they reaches to Golgi. In control siRNA, a CD8M6PR endosome, which are in red, they reaches to Golgi by 60 minutes, whereas in Rufi1 knockdown, they fails to do so. And you can see that a lot of endosomes are still not co-localizing with Golgi. To understand how Rufi1 is regulating the retrograde trafficking of CAM6PR endosomes, we did a tap pull down and found abundant hits of dynein and directin subunits, and we further confirmed the interaction via various biochemical assays to further show that indeed Rufi1 is recruiting dynein on CM6PR endosome with its CD8M6PR assay via live cell imaging and then assess the motility of these endosomes. We found in Rufi1 knockdown, the endosome shows less number of processive tracks and reduced run length, which shows that indeed Rufi1 is regulating dynein recruitment onto these endosomes. Further, we did rescue experiment with wild type rescue construct, and you can see that wild type rescue construct causes the relocalization of M6PR receptor to Golgi, whereas this dynein binding deficient mutant fails to do so. which further confirm our finding that ROFI1 is a dynein adapter for CM6PR endosomes.